All right, well, welcome to Chapter 20. Here we're going to be talking about antimicrobial drugs. And to get us started, we're going to talk about a little bit, very briefly, on the history of chemotherapy. First of all, don't forget, when we say chemotherapy, we're just talking about drugs that are used to treat a disease. So that's the definition of chemotherapy, chemicals that are used or drugs that are used to treat a disease. Remember that an antibiotic is a substance that's produced by a microbe. Usually this microbe will be a fungus, but we will talk about one example where it is actually produced by a bacteria. But it's a substance produced by this microbe that's going to inhibit another microbe. And when we're talking about antibiotics, we're usually talking about inhibiting the growth of bacteria. And of course, these are produced naturally, generally by fungi, to inhibit the growth of this fungus's competitors. So the fungus is producing antibiotics to prevent bacteria from competing with the fungus for its resources. So our natural antibiotics are made this way. Now, one of the first chemotherapy drugs was developed by Paul Ehrlich, and he developed salvarsan, and this drug is used against syphilis in 1910. And this was the first chemotherapy drug, and so the idea of using this chemotherapy to treat microbial diseases was born. And then we've already talked about Alexander Fleming and his discovery of penicillin. He discovered that in 1928 accidentally. Remember, we talked about how the bacteria wasn't growing when he had contamination with that fungus. And but then it still took him 2 decades to purify and test the drug. And then another drug that was important were the sulfa drugs. And they were discovered in 1932 and used for the treatment of gram-positive bacterial infections. So antimicrobial drugs can be classified in a few different ways. They can be categorized based on the spectrum of the antimicrobial drug, as well as how their actions are performed. So when we talk about the spectrum of an antimicrobial drug, we're looking at how many different microbes is it effective against. So a broad spectrum antimicrobial drug would be effective against a wide variety of microbes. So this can be gram-positive, gram-negative microbes, whereas a narrow spectrum antimicrobial drug is going to be more specific to a more specific microbe. When we talk about antimicrobial action, we are considering whether that antimicrobial drug is bacteriostatic or bactericidal. Bacteriostatic is going to prevent or inhibit microbial growth, whereas bactericidal antimicrobial drugs are going to kill those microbes. Other things to consider when considering which antimicrobial drug to use, or when you're considering whether a antimicrobial drug that you're testing is even going to be efficient. So you have to consider where in the body is this antimicrobial drug found, and where is the infection. So for example, if there is infection in the brain, is that antimicrobial drug even capable of passing through the blood-brain barrier to reach the infection in the brain? So these are things that have to be considered. Also, you have to consider the metabolism and excretion of that drug. So is it metabolized and excreted from the body so quickly that it won't even be effective? Again, we already talked about blood-brain barrier. Can this antimicrobial drug survive the stomach acid, or will it be degraded in the stomach acid? As well as what is the half-life of the drug? Those are other considerations. Also, something that's very important to consider is that we need a drug to be selectively toxic. And what this means is that it has to be toxic towards the microbe. And there's lots of things that are toxic towards microbes. Think bleach, for example. But that would be detrimental to us or to the patient that's taking that drug. So it has to be selectively toxic against the microbe, but not against the host. And so drugs can be selectively toxic based on based on the target. So if that target is on the microbe but not on the patient, then that'll work. Also, if that target is more important to a parasite than to the host. So if, say, a certain nutrient is more important to a microbe than it is to us, then you can use that as a drug because that's going to be more detrimental to the microbe than it would be to us. Some drugs can accumulate more in a parasite than it would be in a host, as well as other drugs are only activated in a parasite or in a microbe than it would be to the patient. So next we're going to look at the action of antimicrobial drugs. So there's 5 different actions, and you guys do need to know all 5 of them. But as you see on this slide, there are several examples of drugs for each action, and I don't want you to spend time memorizing the names of the drugs for each action. Just understand the actions themselves. We do go through a few more specific examples on later slides, and you will need to know those examples. But the ones here on this slide, don't worry about memorizing all of these names. So the first action of an antimicrobial drug can be to inhibit cell wall synthesis. And this is a nice, easy target because our eukaryotic cells aren't going to have the cell walls. So that's very effective. Also, inhibition of protein synthesis. This is another great method because remember that bacterial ribosomes are the 70S ribosomes. And so we can target the 70S ribosomes and our eukaryotic ribosomes will not be affected. Another one is inhibition of the nucleic acid. So this can be either an inhibiting replication or transcription. Another target would be the plasma membrane. So some drugs can cause injury to the plasma membrane. So this does pose somewhat of an issue because our plasma membranes are similar to bacterial plasma membranes. And so we usually will only see these drugs that cause injury to the plasma membrane used on superficial wounds. So if we're using them on the skin is already dead. So it doesn't really matter if you are disrupting the plasma membrane on the skin. Also, I think it can be used sometimes as like a last resort, but it is going to cause damage to the patient. And then the last action of antimicrobial drugs is the inhibition of the synthesis of essential metabolites. So here we're going to inhibit certain enzymes that the bacteria has. So that's one of the main ways. So if we inhibit these enzymes, then we inhibit the production of essential metabolites by the microbes. So one of the actions that we just talked about was inhibition of protein synthesis. And I mentioned how this was very useful because we have different ribosomes. And so we're going to look at a few different ways that this happens. So streptomycin is going to change the shape of the 30S portion of the ribosome. Tetracyclines will interfere with the attachment of the tRNA. And chloramphenicol will inhibit the formation of the peptide bond here. So these are a few different antibiotics and how they affect translation of bacteria. Looking at penicillin. So penicillin contains a beta-lactam ring. This is what we're seeing here, highlighted in yellow, is the beta-lactam ring. And that beta-lactam ring is going to prevent the cross-linking of the peptidoglycans. Remember the peptidoglycans that are in the cell wall of bacteria. And so it's going to prevent these from linking together. Here we have a couple of examples of a natural penicillin and what their structures look like. So we have penicillin, and that is going to kill the bacteria by inhibiting their cell walls. Well, bacteria then came back with penicillinase, penicillinase, and that is a beta-lactamase, and it is an enzyme that destroys penicillin. So it takes penicillin, the why can't I say this word? Penicillinase. The penicillinase will then convert penicillin into penicilloic acid. So it's converting it so it's no longer effective. So these bacteria developed resistance. And so then we came back and made semi-synthetic penicillins. And this is just going to add different side chains. So remember the natural penicillins we looked at here. Here's another example here. And you just add different side chains here. And then that is going to be resistant to penicillinase. And so now these bacteria that were resistant to penicillin, now we have the semi-synthetic penicillins, so they're no longer resistant to that. These semi-synthetic penicillins also can be used in a broader spectrum. So the traditional natural penicillin was generally only used for gram-positive bacteria, but now these broader spectrums can also include some gram-negative, can be effective against some gram-negative bacteria. Also, another drug is augmentin, and augmentin combines penicillin with this clavulanic acid, and that is going to inhibit, so the clavulanic acid is going to inhibit beta-lactamase. Remember the beta-lactamase, which is going to break down penicillin. So we're providing penicillin, and we're preventing the bacteria from being able to convert that into the penicilloic acid. And so this is an example of synergism. I believe we've talked about synergism when we use two different drugs simultaneously, and the effects of both of those drugs are going to be more effective than you would if you had just used one of those drugs. Another example of a drug is cephalosporin. And this is very similar to penicillin. It also is going to inhibit the cell wall. But this one is made by the fungus Cephalosporium. And it's actually one of the most common antibiotics used because it is more stable against the beta-lactamases that we've just talked about. It has a broader spectrum, so it can be used for gram-positive and gram-negative bacteria, as well as it's effective against the penicillin-resistant strains. But there is also resistance against cephalosporin, ESBL, and these are gram-negative bacteria that have beta-lactamase enzymes that are able to break down cephalosporin. There's 4 different generations of cephalosporin, and these are categorized based on what they're used for. So the first generation is a narrow spectrum drug used against gram-positive bacteria. The second generation has a broader spectrum, which includes gram-negative bacteria. The third generation is also effective against Pseudomonads, and this is mostly injected, but it can also be taken orally. And the fourth generation is the most, that's the broadest spectrum of all. And the last cell wall inhibitor that we're going to talk about is vancomycin. So this one is a glycopeptide that is produced by Streptomyces. And this is a bacteria. Again, it's going to inhibit the cell wall, and it's used to kill MRSA. This is a antibiotic resistant strain. So we used to use vancomycin against these antibiotic resistant strains, but then resistance against vancomycin has also been developed. And so then we treat this with Cinercide. This is going to block protein synthesis, but it's extremely expensive and has risky side effects. But you can kind of see this similar story where we develop a new drug and bacteria develops resistance, and this just keeps going, right? So I was talking about antibiotics. And as we move forward to start looking more into the viral and eukaryotic drugs, it's important to keep in mind that this is a much more challenging feat. So remember, bacteria and the drugs that we used against them, they're easier to develop because bacteria have a lot of different structures than our eukaryotic cells have. So remember, we were talking about the targeting of the cell wall, how bacteria have different ribosomal structures, how bacteria have different metabolic activities. So these are all very useful targets that we can use against bacteria. But when we start looking at like funguses, protozoans, and helminths, they're eukaryotes just like we are. And so it's much more difficult to find a drug that is going to be effective against them and not us. Also, viruses present a whole new set of challenges because remember, first of all, they're not something that's living. So you can't kill something that's not alive. Also, they are often going to be hiding inside of cells. And so this presents a whole new challenge too. So generally here, we're going to have to disrupt some form of their life cycle to prevent them from replicating. But as far as completely inhibiting all virus, that's very difficult. So first we'll look at antifungal drugs. Polyenes, such as nystatin, they're going to inhibit ergosterol. So ergosterol is inside of fungal cell membranes. It's similar to the cholesterol that we have. So this is one lucky thing that a lot of, or most funguses actually have. So we can target this ergosterol that we do not have because we have cholesterol instead. So a lot of your fungicidals will target ergosterol. Another example is griseofulin. And this is from penicillin. Penicillin is going to be producing it. And it's going to block the microtubule assembly during mitosis. And this is what we used against the tinea or ringworm. Now looking at viruses, so some of the modes of action of antiviral drugs include blocking the receptors on the host cell. So remember, in order for a virus to enter, it has to bind to the receptor on the host cell. So if we block those, and we block the virus from even entering the cell to begin with, we can prevent viral uncoating, inhibit viral DNA integration into the host genome, inhibit nucleic acid synthesis, and block viral budding. A couple other drugs, so imiquimod is going to promote interferon production. So if you recall interferons and how they have their antiviral properties, this drug is going to encourage the body to promote more interferons, which is very useful. And then another example is antiretrovirals. So if you remember reverse transcriptase, remember that's the enzyme that converts RNA into DNA that the retroviruses have. So if we inhibit that enzyme, then you're going to inhibit the life cycle of those retroviruses. One example of an antiviral drug is acyclovir. And this is you'll see this drug used a lot against the herpes viruses. And what it is a nucleosidase analog. So what this means is it looks very similar to the nucleosidase deoxyguanosine. So here's deoxyguanosine, and here's acyclovir, and they look very similar. And so what's going to happen is the acyclovir will be incorporated into DNA, but then the DNA will not be effective. So that's going to block the DNA synthesis of the herpes virus. But what's kind of cool about this is the acyclovir is only converted into the nucleotide analog inside virus infected cells. So this is not going to affect cells that are uninfected. Then looking at antiprotozoan drugs. So some examples are chloroquine is effective against malaria, and that's going to accumulate in the pathogen's food vacuole and form toxic compounds that are going to kill the cell. Quinacrine is effective against Giardia, and it's going to inhibit the transcription and translation of it. Metronidazole is effective against Trichomonas vaginalis, or also some anaerobic bacteria. And this is selectively activated in the target cells, and it will inhibit the DNA synthesis. Some examples of antihelminthic drugs include praziquantel. This is effective against tapeworms. This will alter the cell membrane, and by altering the cell membrane, it's going to expose these antigens so that the host immune system can then attack them. Also, mebendazole. This is a broad spectrum antihelminthic drug, and it inhibits nutrient absorption. And lastly, ivermectin, and this is effective against nematodes, mites, lice, and ticks. And this is going to selectively alter the intervertebrate cell membrane, their cell membrane permeability, and that will cause death of the parasite. Now, some of these organisms will develop resistance against these drugs. So they can mutate the target gene. They can increase production of the target. They can decrease the drug accumulation by excreting the drug, or they can inactivate the drug. So antibiotic resistance, as you saw, is a big problem. And it is developed through mutations, or also it can be transferred horizontally through plasmids. We've talked about those two methods. Mechanisms of antibiotic resistance. So some of the most common ones are enzymatic destruction of the drug. So we already saw that example where the enzyme is going to convert the drug into a different form, where then it's no longer effective. The microbe can also prevent the penetration of the drug from even getting in to begin with. The microbe can alter the target site so that the drug no longer can use that as a target. And it can also rapidly eject the drug. This here is a graph of antibiotic resistance. So what we're seeing on the black line is bacterial numbers. and the red line is antibiotic resistance. So over time, once antibiotic is administered, bacterial numbers will decrease until antibiotic resistance is developed. And then those numbers will then come back because these bacteria are now resistant to the antibiotic that is being used. These are the same forms, but in picture form. So first mechanism, blocking the entry. Second one is inactivating these enzymes. These enzymes that are going to inactivate the antibiotic. Also altering the target molecule and efflux of the antibiotic just by pumping it back out. Antibiotic resistance is encouraged by the misuse of antibiotics. And misuse can include things like using outdated and weakened antibiotics, or by using antibiotics for viral infections, or by using antibiotics for prevention. So we see this a lot. Sometimes they'll give animals antibiotics just before they're even ill or have an infection, just to prevent them from having it. But of course, that's going to lead to antibiotic resistance. Also, if you fail to complete the prescribed regimen, so some people will feel better when they've been taking antibiotics, and they'll be like, well, I feel better, I'm not going to take the drugs anymore. But that doesn't, just because you feel better doesn't necessarily mean that all of the bacteria are dead. So that's the ones that are still alive or the strongest ones. So of course, they're going to then grow back with some resistance. Also, using someone else's leftover prescription is considered misuse of antibiotics. And just as a quick reminder of how this works, so if you're initially infected, these are all of the bacteria, but the two little red guys are the resistant ones. And then you take antibiotics, but you don't take them completely, or maybe you do, and the resistant ones survive. Then afterwards, you've killed off all your good bacteria, and then now you're just left with the resistant bacteria, which will come back stronger than ever because now they don't have the competition. And so that is how natural selection plays a role in the development of antibiotic resistance. And that is going to complete our lecture on chapter 20. And I will see you guys soon for chapter 7.